Today we're going to talk about how traits are inherited, and the goal of this video is to define a trait and define an allele, and to describe what determines whether a trait will be passed to an organism's offspring and how different combinations of alleles result in different traits. That's a lot of ground to cover, so let's get started. First, what is a trait? A trait is a specific characteristic of an organism. This would be things like the organism's eye color or height or flower color. And traits are controlled by one or more genes. Gregor Mendel, who lived in the 1800s, was an Austrian monk who studied the inheritance of traits in pea plants, and he's commonly regarded as the father of genetics because his work showed that traits were inherited in predictable ways. So I want to take a couple of minutes and try to summarize Mendel's experiments. Mendel studied 7 traits in pea plants. He studied the seed form, which could be round or wrinkled, the seed color, which could be yellow or green, the pod form, which could be inflated or constricted, the pod color could be green or yellow, the flower color could be purple or white, the flower position could be axial or terminal, and the stem length could be tall or short. So those were the 7 traits that he studied in pea plants. In this video, I'm going to focus on the seed form and the flower color. So let's start with the seed form. He started his experiment with what's called true breeding plants, and a true breeding organism produces offspring with the same trait as the parents. So you'll notice that we've got wrinkled and round here as the 2 types of seed form. So a plant that produces all wrinkled offspring would be a true breeding wrinkled plant, and a plant that produces all round seeded offspring is a true breeding round pea plant. Now, to get a true breeding plant, Mendel used a method called self-pollination. And self-pollination is basically where you take the pollen from a plant and you just let that pollen fertilize its own eggs. And you can do that with a brush. You just put some pollen on the tip of a paintbrush and rub the paintbrush on this part of the plant, and the pollen travels down and fertilizes the eggs, and the plant will self-pollinate. Okay, once he had these true breeding plants, he crossed a true bred round pea plant with a true bred wrinkled pea plant. And the way that you do that, instead of letting the pollen from the plant fertilize itself, you just use a paintbrush and take some pollen from one plant and put it on the other plant. That's how you cross pollinate. So that's what he did here. He just crossed a true bred round pea plant with a true bred wrinkled pea plant. Now, what did he observe? So out of the thousands of the seeds that were produced, all of them were round. Every single one of them. None of them were wrinkled. So that's what you see here. These represent the parents. You've got a wrinkled parent and a round parent. And all the offspring were round. So he then took 2 of the offspring, let's say this one and this one, and he crossed them. Here's what he observed. In that next generation of offspring, the wrinkled form, the wrinkled seed, reappeared. So this is now the second generation, and the wrinkled form reappeared there. It wasn't in the first generation, but it was in the second. And he saw these same results for the other 6 traits that I mentioned a couple of slides ago. So what did Mendel conclude from all this? He said that traits are determined by a unit. That was his term, a unit that's passed from parent to offspring. So he's saying there's something that's getting passed from parents to offspring, and I'm calling that a unit. Now, in Mendel's time, remember this was the 1800s, DNA hadn't been discovered yet. We now know that what he was really referring to were genes. The individual inherits a gene for each trait from both parents. So every pea plant has 2 genes for a trait. And Mendel said that traits may not physically appear in an individual, but the genes for them, what he called the units, can still be passed to the offspring. So that's what was happening here. Even though in this first generation, the wrinkled trait didn't appear, this first generation, they still carried the gene for the wrinkled form, and that's what got passed on to their offspring down here. Now, with the remainder of the video, I want to try and show you how Mendel's work has been expanded upon. So we now know that in sexually reproducing organisms, chromosomes are found in pairs, and each parent contributes one chromosome to the pair. So for example, here's a sperm cell. Inside that sperm, you have in this example, there are 2 chromosomes. Here's an egg cell. Inside the egg cell, there are 2 chromosomes. When the sperm fertilizes the egg, those chromosomes pair up. And the pair is called a homologous pair. So this is a homologous pair, and this is a homologous pair. Now, each member of that pair we call a homologue. So each homologue has the same arrangement of genes for a specific trait. So you'll notice this is a homologous pair. They're different colors because one of them comes from the father and the other comes from the mother. So it's just a way of distinguishing them. But notice these bands going across them, those represent genes for a specific trait. And the bands are all lined up because the genes are arranged in the same way within the homologous pair. So each homologue has the same arrangement of genes for a specific trait, but the alleles, which is a version of a gene, those can differ on each homologue. And I'll say more about that in a moment. So I'm going to focus now on the example of flower color in Mendel's experiment, but I'm going to explain it with what we now know from experiments that have been done after Mendel. So inside of a cell's nucleus, there are pairs of homologous chromosomes. And every homologous pair consists of 2 chromosomes. One is paternal, meaning it comes from the male parent, and one is maternal, meaning it comes from the female parent. And at a certain position, which is called a locus, on each of these homologues, so here's a homologue, here's a homologue, at a certain position, that's this light brown band, at a certain position, called a locus, is a gene for a trait. So this represents the gene for flower color on this homologue, and here we have the gene for flower color on this other homologue. Now, you'll notice these genes, they're different colors because they represent different alleles. There are different versions of a gene, and each version is called an allele. This entire science table on its face on the floor. So there are different versions of a gene, and each version is called an allele. One of the alleles codes for white flower color, and the other allele codes for purple flower color. So this green band here, it represents the allele for white flower color, and this one represents the allele for purple flower color. An allele can be dominant or recessive. The dominant allele expresses the trait that's associated with it, and it masks the trait that's associated with the recessive allele. And the recessive allele can only express the trait that's associated with it when the dominant allele is absent. Now, that will become more clear in a moment on the next slide. You usually represent the dominant allele with an uppercase letter and the recessive allele with a lowercase letter. So in our example of flower color, there's 3 possible combinations of alleles. In this one, we have This one would be homozygous dominant, and that's because there are two dominant alleles, big P, big P. Then there's heterozygous. That's where you have one of each. So you have a dominant allele and a recessive allele, a big P and a little p. And then there's homozygous recessive, which would be two recessive alleles, little p, little p. Homozygous, that word homozygous just means both alleles at that locus are the same. So this one's homozygous because both of these are the same. This one is homozygous because both of these are the same. Heterozygous means that the alleles at that locus are different. So you have one dominant, one recessive, a big P and a little p. So those are the three possible combinations of alleles for flower color. Now we're going to finish up by talking about genotype and phenotype. So on the left here, you'll see the combinations of alleles that we were just talking about. Big P, big P, big P, little p, little p, little p. Now, when we say that, we're talking about a genotype. When we say the combination of alleles, we're talking about a genotype. And so there's three possible genotypes here. There's the homozygous dominant genotype, the heterozygous genotype, and the homozygous recessive genotype. Phenotype is the physical characteristic that you observe. So in our example, the phenotype, there's only two. There would be purple flower color and white flower color. Those are the two possible phenotypes. So you'll notice, going back to the terms dominant and recessive, whenever a dominant allele is present, purple shows up. But when a dominant allele is not present, like down here, white shows up. So the dominant allele, in this case, look at the heterozygote, the flower that's got big P, little p. The dominant allele masks the trait that's associated with the recessive allele. The flower still turns out to be purple, even though it carries the allele for white flower color. And that's because purple is dominant in this case.